Assalamu alaikum everyone. Guess we will start now, okay? So Assalamu alaikum everyone. Just hold my camera. Okay. Assalamu alaikum. I'm Abdul Razzak Cham. I'm a second year medical student, and today I'm going to be explaining the blood gas transport lecture. So first of all, this is going to be a very easy lecture, inshallah. It's going to be straight to the point. You even took some of the ideas here in Moldwan with Dr. Hannah. Okay, so if you have any questions, you can contact me either through my email or through my phone number, whatever you are comfortable with. With that being said, we can start now. So if you have any questions, you can either use your mic or type in the chat, or if you want me to repeat any idea. Are we good for now? Okay, so bismillah. So first of all, the first idea of this lecture is that we need to understand that there are two forms of transportation of oxygen: the physically dissolved form and the chemically bound form. The physically dissolved form is basically the free oxygen that's moving freely inside the blood. When these free molecules of oxygen move, they are going to produce pressure. Okay. What about the chemically bound? The chemically bound is basically the oxygen molecules that are bound to hemoglobin. So in this graph here, the hemoglobin was removed in this example, and we are discussing only physically dissolved for this example only. Okay. So this graph shows the partial pressure against the O2 concentration. And this line here shows the amount of physically dissolved oxygen. As you can see here, and according to Henry's law, an increase in PO2 is going to cause an increase in the concentration of O2. This is not CO2, this is the concentration of oxygen, okay? So as partial pressure increases, you are going to see an increase in O2 concentration. You need to know that as much as you can increase partial pressure, O2 concentration is going to keep increasing in the physically dissolved form. The chemically bound form has another case. We're going to discuss that later. Now, for the partial pressure, as you keep increasing the partial pressure, O2 concentration will just keep increasing up until infinity. Okay, but as you can see here, the O2, the physically dissolved form of O2 is not enough to provide adequate oxygenation to the organs. Therefore, we're going to need another form for for oxygen transportation, and that's where the chemically bound form comes. Okay. Okay. So in this graph here, we can see the physically dissolved line again. This is just the same as this line. Okay. But now we can see a non-linear graph too. This graph here shows the total concentration of O2. This includes both the physically dissolved and the chemically bound. Okay, so you can tell that the chemically bound is the difference between the total oxygen and the physically dissolved. That's simple because if you have a total amount of oxygen, then you remove the physically dissolved. All the remaining is going to be chemically bound. So as you can see here, as you increase partial pressure, the difference between the two graphs are going to increase. And what did we say? The difference between the two graphs was, it was the chemically bound. Therefore, you can tell that the amount of chemically bound is going to increase as you increase the partial pressure. You can see that this will continue up until a limit. At the partial pressure of 100 here, you can see that the difference between the two graphs, the total oxygen and the physically dissolved, is going to become constant, which means that the amount of chemically bound is going to become constant. Why does that happen? It basically happens because at that point, all the possible binding sites for oxygen are full. There are no places for oxygen to bind. Okay. At that point, we can say that we have reached O2 capacity. Again, at O2 capacity, what does that mean? It means that all the possible binding sites for oxygen on hemoglobin are occupied with oxygen. So any additional oxygen that's going to come is going to be in the physically dissolved form. So at that point and on, you can see that both graphs are going to increase at the same rate, or you can just say that they are like moving in parallel. Okay, which means that all the increase is going to be for the physically dissolved. Again, you need to know a very important concept, that O2 capacity only includes the chemically bound. It has nothing to do with the physically dissolved. Okay. Something before we move on. is that as you can see here, as much as you keep increasing partial pressure of O2, the physically dissolved is going to keep increasing, which you just said before. Okay, but chemically bound has a limit at about partial pressure of 100, where saturation is going to be reached and all the binding sites for oxygen are going to be full. Okay. Now we're going to discuss some concepts about equilibrium. As you can see here, compartment A and compartment B both have the same amount of free O2 molecules. Remember when I said that these free O2 molecules are going to produce pressure when they are going to move? So you can tell that the amount of pressure produced on both sides is going to be equal because both of them have the same amount of oxygen. Okay? So we can tell that PO2 on the A compartment is equal to the PO2 in the B compartment. What about the next picture? In the next picture here, we can see that hemoglobin was added to this compartment of the chamber, okay? So what happens here is that hemoglobin is going to bind molecule because that is function, okay? So now we are going to have less free O2 molecules. So something about chemically bound molecules that's not present in the physically dissolved is that chemically bound cannot produce pressure. You can think of it like because chemically bound are bound to hemoglobin, they cannot move, therefore they cannot produce pressure. So only the free O2 molecules are able to produce pressure. So if we compare the amount of pressure produced on compartment A with the amount of pressure produced by only these two free molecules in compartment B, you will find that the pressure in compartment A is greater than the pressure in compartment B. And due to a pressure gradient, O2 is going to diffuse from this side to this side, just like anything. Remember in the previous lecture when we took that O2 is going to diffuse from the alveoli to the capillary, okay? This is exactly the same thing because PO2 on this side is more than PO2 on that side. O2 molecules are going to diffuse from this side to the other side, okay? Okay, so because more O2 molecules diffuse from this side from A to B, now hemoglobin is going to bind more molecules. It won't say no for more molecules, okay? Now you can see that there are two free molecules on the A and two free molecules on B. Therefore, we can tell that equilibrium is reached between the two compartments. Again, because the chemically bound cannot produce pressure. Only the free dissolved O2 molecules can produce pressure. So PO2 on A is equal to PO2 on B. And a very important concept that we need to know, guys, is that there is also an equilibrium, not between the two compartments, but there's also equilibrium between the chemically bound and the physically dissolved. Which means that when one physically dissolved molecule leaves or diffuses away, the hemoglobin is going to leave an O2 molecule instead of it. It's going to like sort of replace it, okay? On the other hand, if an O2 molecule diffuses in, the hemoglobin is going to bind it, okay? So it's going to take up more oxygen molecules. Again, if an O2 molecule diffuses away, the hemoglobin is going to leave a molecule instead of it. But if more O2 molecules came toward the hemoglobin is going to bind more molecules, okay? So one last thing is that although you can see that this concentration or the amount of oxygen, the amount of oxygen on this side is about 14 molecules, but here it's 2. Why wouldn't oxygen flow from this side to this side? That's basically because this is a difference of concentration. However, the pressure on both sides is equal. And we said that the driving force for oxygen movement is pressure. It is not concentration. Therefore, oxygen won't flow on this side. You can see that the amount of the chemically bound oxygen is a lot larger than the physically dissolved. And we said that the amount of physically dissolved is not enough to provide adequate oxygenation. It's not enough to it's not going to provide adequate oxygenation to the organs of the body, okay? Therefore, however, that doesn't make the physically dissolved oxygen useless. It's basically the physically dissolved form is very important because it is the only form of oxygen that can diffuse through barriers. So the chemically bound is bound to hemoglobin molecules. Therefore, it cannot cross the barriers. So it can't cross from the alveoli to the capillary, or from the capillary to the tissue. Only the physically dissolved can cross the barriers. That's the importance of the physically dissolved type of molecules, okay? Someone is asking that I said an oxygen molecule leaves, hemoglobin leaves with it too. No, what happens is that, I might have said that by mistake, but what basically happens is that if this molecule, for example, left or diffused away, hemoglobin is going to leave a molecule, an O2 molecule, to replace it, okay? On the other hand, if an O2 molecule came here, okay, so there are going to be more O2 molecules around the hemoglobin. Hemoglobin is going to bind that extra molecule of O2. Okay, this will continue up until a capacity where all the hemoglobin molecules are filled with oxygen, which we said that here we have reached like O2 capacity, okay? But again, for your question, when an O2 molecule diffuses away, the hemoglobin molecule is going to leave a molecule. It's basically it has like a molecule bound to it. Now it's going to leave it, so that molecule is going to replace it. Got it? Okay, good. So do you have any questions up till now? Should I repeat anything? Okay, so I guess we're good. Okay, now this is the O2 binding curve. Okay, the O2 binding curve, or also called the O2 dissociation curve, it shows the O2 saturation, or the percentage of the O2 binding sites that are actually occupied with oxygen. So let's just you know that each hemoglobin molecules has four binding sites. Okay, so let's just imagine that the body has like 100 available binding sites that oxygen can bind to. When all of these binding sites are occupied with oxygen, we are going to have a saturation of 1 or 100% saturation. I don't know if you know from like movies or stuff, when they say that oxygen saturation is 100 or 95 or something, that's basically it basically means that all the binding sites are occupied with oxygen. Okay. So on this graph, we have to know 3 main points. The first one is the P50 point, or the P0.5. The P0.5 or the P50 is basically the partial pressure of oxygen at which the oxygen saturation is 0.5. Okay. This point is very important because we are going to use it to know if we have a shift to the left or a shift to the right. We're going to discuss what the shift to the left means and what the shift to the right means later. Okay. But for now, you have to know that a P50 is the partial pressure required to have a saturation of 0.5 or 50%. Which point exactly? The P50 or the saturation thing? The P50. Okay, I'm going to repeat it. Okay. Okay, so basically, the first important point that we're going to use is P50. P50 is the point of partial pressure, okay, at which we have a saturation of 0.5, as you can see here. What does a saturation of 0.5 means? It means that 50% of all the binding sites of oxygen are actually occupied with oxygen. Okay, so this point is very important. Usually, we have a 50% saturation at a partial pressure of 27 millimeter mercury. So you need to know this 27 because basically we are going to use this point to know if we have a shift to the left or a shift to the right. So is it clear? So simply, B50 is the partial pressure at which we have 50% saturation. Okay? Okay. The second point that we need to know is the point at 100 millimeter mercury. So at the point of 100 millimeter mercury, we are going to have a 0.98 saturation or 98% saturation. Where can we find this 100 millimeter mercury? We basically find it, remember in the previous lecture where in the alveolar capillary, we have the blood comes and CO2 leaves and O2 comes. And at the end of the capillary, we have the most oxygenated blood of our body. That blood there is going to have a PO2 of 100 millimeter mercury and a saturation of 0.98. Okay. The third point is the point of 40 millimeter mercury, where we would have a saturation of 0.75. At the 40 millimeter mercury, where can we find 40 millimeter mercury's partial pressure? It's actually in the same place here, but at the beginning of the capillary. When mixed venous blood comes, it's going to have a partial pressure of about 40, and it's going to have a saturation of 0.75. Okay. So again, so the mixed venous blood that comes from the heart to the alveoli is going to have a PO2 of 40% and an oxygen saturation of 0.75. When the exchange happens and the blood is about to leave, okay, it's going to have a PO2 of 100 and saturation of 0.98. Okay. You might argue that 0.75 is a very high percentage, and why does the blood needs to get reoxygenated although it has this amount of oxygenation? It's basically because this is mixed venous blood, and mixed venous blood is blood that's combined from organs from all over the body. So blood is coming from the heart, it's coming from skeletal muscle, from the kidneys, and so on. So blood that's coming back from the heart might have a saturation of about 0.4, for example, and the blood that's coming back from skeletal muscle might have a saturation of 0.8. So some organs are going to consume more oxygen than others, and are going to bring blood that's less oxygenated than other organs. But when all the blood from all organs combine, it's going to give an oxygenation of 0.75 or a saturation of 0.75. Okay. And so we need to reoxygenate this blood to provide the enough oxygen to the organs that require a higher demand of oxygen. Okay. So Dr. Shams here wants you to understand the difference between these two graphs. Okay. So this graph here shows the O2 concentration in relation to the partial pressure, and this graph here shows the O2 saturation in relation to partial pressure. So a very important point that Dr. Shams wants you to understand is that when PO2 decreases, we are going to see a decrease in both the O2 concentration and O2 saturation, which is simple. You can easily visualize that on the graph. As PO2 decreases, you are going to see a decrease in both the concentration and the saturation. But a very tricky concept, other than this one, is the fact that when we have anemia, in cases of anemia, and when we lose a portion of our hemoglobin, what happens is that we are going to see a decrease in concentration, but saturation is going to stay the same. I'm going to explain why. Okay, so let's imagine now that we have 6 hemoglobin molecules, okay? Okay, and only for this example, each hemoglobin molecule can only bind 1 O2, okay? And this is only for the sake of simplicity. In real life, each hemoglobin can bind 4, okay? But when all these binding sites are full with oxygen, we are basically going to have a saturation of 1, okay? 100% of the possible binding sites are occupied. But what about concentration? If we calculate the concentration, we can say that it is 6. We have only 6 O2 molecules bound. Okay. What happens in the case of anemia? That we are going to lose some hemoglobin. Let's just say we have a 50% anemia and half of the hemoglobin is lost. Okay. In that case, we are only going to have 3 binding sites for hemoglobin for O2, but all of them are occupied. So we are still going to have 100% saturation. Now we're going to forget about these binding sites because they no longer exist. Only these are going to be counted. Therefore, when all of them are occupied, we're going to have a saturation of 1. Okay, that's it for saturation. What about concentration? If we count now the O2 molecules that we have, we're going to have a concentration of 3. Okay, not 6. So we can tell that concentration is halved. So in case of 50% anemia or anemia in general, saturation is not going to get affected. In the case of a 50% anemia, concentration is going to be halved, okay? Because now we are going to have like half of the amount of oxygen is like cannot be bound down. Okay, so now concentration is going to be halved. Okay, so do you have any questions, guys, for now, or do you want me to repeat anything? Okay, guess we're good. Right. Okay, now we are going to discuss the shifts in the O2 dissociation curve. Okay, and what factors can cause these shifts, either to the left or to the right. Okay, first of all, this is the normal graph that we just discussed, the red one. Okay, we can see that we have a B50 here. What's B? Oh, sorry, B50 is here. Okay, B50 is at about 27, like we just mentioned. Okay, the green graph is a shift to the left. Okay, if we looked at P50 in of the green graph, we can see that P50 here is what? It's 20. Okay, or about 20. Okay, so P50 is again is the pressure at which we have 50% saturation. So if we locate the saturation of 50 here, we're going to find that the pressure is about 20. Okay, what does a decrease in PO2 mean? Oh, sorry, in P50 means? It means that now you can reach that point with a less PO2, which means that there is an increase in affinity. Again, so a shift to the left means that there is an increase in affinity, and now you can reach P50 with a lower PO2. What does a shift to the left mean? To the right means? Sorry, a shift to the right means that there is a decrease in affinity, and now we require more pressure to reach a 50% saturation. Is it clear? Okay. Okay, now we're going to discuss the first two factors that are going to affect the oxygen dissociation curve. Okay, so the first one is pH. As you can see here, in case of acidosis or a decrease in pH, what happens, what causes a decrease in pH is an increase of H plus ions, okay, or hydrogen ions. So in case of increase in hydrogen ions, you are going to see a decrease in pH or acidosis. In that case, as you can see here, you can see a shift to the right, okay? Which means we have a decrease in affinity. How does that happen? That basically happens by when H plus increases in blood, it's going to go and bind to hemoglobin. And it's going to cause conformational changes, like changes in the shape of the hemoglobin, and cause the binding of O2 to the binding site to be more difficult. Okay, basically it's going to decrease the affinity by acting directly on the shape of the hemoglobin molecule. Okay, this effect by H plus on hemoglobin is known as the Bohr effect, BOHR. We're going to talk more about Bohr effect in the next slide. Okay, what about in case of alkalosis? In case of alkalosis or an increase of pH, we're going to see a shift to the left. A shift to the left means again that there is an increase of affinity. How does that happen? Basically, in case of alkalosis, you're going to have less H plus in blood. And when you have less H plus in blood, we're going to have less H plus binding to hemoglobin, okay? And when you have less H plus binding to hemoglobin, the affinity of hemoglobin to O2 is going to increase. That's it about the pH, okay? Now let's go to CO2. For CO2, as you can see here, in case of hypercapnia or an increase in CO2, PCO2, you can see that we have a shift to the right. How does that happen? It basically happens in two ways. So CO2, when it increases, it has two mechanisms to cause a shift to the right or decrease affinity. The first one is a direct effect of CO2 on hemoglobin. Basically, it's going to go and bind to hemoglobin and cause it to have less affinity to hemoglobin. Basically, it's going to form something called carbamino hemoglobin. And this carbamino hemoglobin has less affinity to O2. The other indirect way happens when CO2 interacts with what? With water. Water is present everywhere in our body. Okay? So when CO2 interacts with water, it's going to produce H plus ions plus HCO3 minus or bicarbonate. So if you can remember from 1 minute ago, what does H plus happens when it increases in blood? It's going to go and bind to hemoglobin and do the Bohr effect, causing a decrease in affinity. Again, so CO2 has two mechanisms to decrease affinity. The first one is by acting directly on the hemoglobin molecule, and the second one is by interacting with water, producing hydrogen ions, and the hydrogen ions by its turn is going to decrease the affinity of hemoglobin by binding to it. Okay? Okay. So why did I include both of these effects on the same slide? Basically because the effects of these two are combined. Usually, hypercapnia or an increase in CO2 is associated with a decrease in pH, okay? Or an increase in H plus ions. And hypocapnia is usually, most of the times, associated with an increase in pH or a decrease in H plus ions. And we just discussed how. We just discussed that CO2 plus H2O are going to give what? H plus. And this H plus is going to cause this alkalosis that's decreasing the pH. So are we good for pH and CO2? Should I repeat anything? Okay. Now let's move on to the Bohr effect. I just explained it quickly that H plus binds to hemoglobin and it's going to cause some shape changes to the heme. What is heme? Heme is basically where the site where O2 is going to bind to hemoglobin. So it's going to cause some shape changes in heme, therefore making the binding of O2 to the heme more difficult. That's basically the Bohr effect. However, this effect is very important because it has very important implications in our bodies. The first one is in the alveoli. So you can see that the mixed venous blood comes here, okay, and it has CO2. Okay, now CO2 is going to diffuse from the capillary to the alveoli. Why? Due to a pressure gradient. This was yesterday's lecture, I guess. Okay, so CO2 is going to diffuse from the capillary to the alveoli due to a pressure gradient. And when CO2 does that, the hemoglobin here is going to have a higher affinity to oxygen. So it's going to bind the diffusing O2 from the alveoli to the capillary with a higher affinity. Why does O2 diffuse from the alveoli to the capillary? Again, due to a pressure gradient. Okay? Again, so CO2 is going to leave the alveoli. Now hemoglobin here is going to have a higher affinity to O2 and is going to bind O2 with a higher affinity. Okay? The second implication of Bohr effect is seen here at the tissue level. So let's just say this is a skeletal muscle, for example, skeletal muscle, okay? And it's producing CO2. CO2 is produced in the skeletal muscle as a result of like its normal functions, such as the Krebs cycle, cellular respiration, and this stuff, okay? So when oxygenated blood comes here, the CO2 is going to diffuse from the tissue to the capillary. Why? Due to a pressure gradient. Therefore, it is going to cause a decrease in the affinity of the hemoglobin here. And this hemoglobin, because now it has less affinity to O2, is going to release more O2 to the tissue. Okay, it's going to release O2 to the tissue. Okay, that's basically the implication of Bohr effect. Dr. Hashim explained the third example, which is like a combination of both the alveoli one and the tissue one. So I'm going to go over it a little bit quickly. Okay, so this is the maternal blood, and this is the fetal blood. Okay. So let's start from the fetal side. So the blood of the fetus has CO2, okay? And CO2 is going to diffuse to the mother's blood due to a pressure gradient. And now hemoglobin here of the fetus is going to have a higher affinity to oxygen. So it's going to bind oxygen that's coming from the mother with a higher affinity. What about the blood of the mother? So hemoglobin of the mother will come with O2, okay? And because of this CO2, that is given by the baby to the mother, the hemoglobin here is going to have a lower affinity to O2. So basically now the hemoglobin here is going to release more O2 to the fetus, okay? So you can think of the fetus as the alveoli, as the blood in the alveoli that wants to give out CO2 and take O2, and you can think of the maternal blood as the blood of the tissue that is taking CO2 from the baby and giving out O2 in exchange of it. Which idea? The placenta one about the baby and the mother, or the whole thing? Okay, okay, okay. Let me erase this. Okay, so a blood from the baby is going to come and it's going to have CO2. Okay, because the baby cannot like exhale, it's going to give out its CO2 to the mother. Okay, now this CO2 is going to diffuse from the baby's blood to the mother's blood. Why? Due to a pressure gradient. Okay, let's forget about CO2, this CO2 for a while. Okay, that has diffused. Let's focus on the hemoglobin of the baby. Okay, so because CO2 has left the circulation of the baby, now the hemoglobin here is going to have what? A higher affinity to hemoglobin, to O2, sorry. Okay, so hemoglobin here is going to have a higher affinity to O2. And because it has a higher affinity to O2, it is going to bind the oxygen that's coming from the mother with a higher affinity. Okay, what about you can think of it as the blood in the alveoli, where it's going to give CO2 and take O2 with a higher affinity from the alveoli. But now it's taking it from the mother. That's the only difference, okay? What about the maternal blood? So maternal blood is going to come and is going to have O2 or oxygen, okay? Because of the CO2 that was released by the fetus to the mother, now the hemoglobin here of the mother is going to have a less affinity to what? To O2. Therefore, it is going to release even more O2 to the baby. You can think of it as the blood of the tissue here. Here, the blood is getting its CO2 from the tissue, but in this example here, the blood is getting the CO2 from the baby. Okay, and it's giving out the O2 here to the baby instead of giving it out to the skeletal muscle. Okay, is it clear? Okay. Yeah, it's going to have less affinity because CO2 has came from the baby. Okay. Tamam. Okay. Now we're going to discuss the third factor that's going to affect the dissociation curve. It's temperature. So basically, you need to memorize these because there is not a way to understand them. It's more like memorization. Okay. But basically, an increase in temperature is going to cause a shift to the right or a decrease in affinity. And on the other hand, a decrease in temperature is going to cause a shift to the left or an increase in affinity. One implication of this is when you are training and you want, and when you're training and the muscle is producing heat, basically producing heat energy. Okay. 1 second. Okay. Sorry for that. Okay. Now the skeletal muscle is producing heat. It's producing temperature, so that when the blood comes and it wants to supply it with O2, the blood is going to get affected by the heat produced by this muscle. And the hemoglobin here is going to have what? It is going to have less affinity. Again, because temperature will cause a shift to the left and a decrease in affinity. So because hemoglobin here has a less affinity to oxygen, now it is going to even release more and more O2 to the skeletal muscle. And this skeletal muscle really needs that O2 because it's going to use it because it's exercising. Okay, so this muscle requires more O2, and through this mechanism, through the production of heat, it's going to decrease the affinity of hemoglobin to O2, and more O2 is going to be released to the skeletal muscle. Okay, the fourth factor is the BBG, or biphosphoglycerate, or also called diphosphoglycerate. Okay, this BBG, when it increases, it's going to cause a shift to the right and or a decrease in affinity, and when it decreases, it's going to cause a shift to the left or an increase in affinity. When does that happen? So basically, the only implication of this and the only example for that is that in cases of chronic hypoxia, such as when you go up high in the mountains, or in cases of chronic lung diseases, in that case, you are going to see that there is an increase in BBG, okay? And this increase in BBG is going to cause a decrease in affinity. Okay, why is that helpful? It's helpful because this decrease in affinity is going to assist in the dropping of oxygen at the peripheral organs, so that peripheral organs are not going to suffer from hypoxia or decrease in of O2 in cases of chronic hypoxia, okay? They're basically going to still get their adequate oxygenation, even though the body is going is in the case in a case of chronic hypoxia. Okay. Okay. This is like a summary slide. I'm going to go it really quickly. Okay. So first of all, we have CO2 and H plus. When both of these increase, they are going to cause a shift to the right. And we said that an increase in CO2 is most of the time associated with an increase of H plus. The third factor is temperature. Okay, when temperature increases, we are going to see a shift to the right or a decrease in affinity. The last one is diphosphoglycerate, and it's also going to cause a decrease in affinity. Okay, how can you memorize this in a better way? I think Dr. Hana gave you this mnemonic, but I'm going to repeat it now. Basically, CO2, we can write a C. An increase in H plus or a decrease in pH is basically an example of acidity or a case of acidity. So we're going to write A. Then diphosphoglycerate or biphosphoglycerate, we're going to use the letter D, and we're going to write the D here. Okay. Then we have an E, and this stands for exercise. You don't have to know it for this lecture, but it's only for your information. Okay. The last one is temperature. Okay. So this cadet faces right. So you can understand from this mnemonic that when carbon dioxide increases, or when we have acidity, when diphosphoglycerate increases, or when we have an increase in temperature, these factors are going to cause a shift to the right or a decrease in affinity. Okay, so this summarizes the whole last 4 slides. You guys need to be really good with these ideas because these are very important, and most of the questions about this lecture are going to be about the shift to the left or shift to the right. Okay. Okay. Now we are going to discuss 2 cases in which we have an inactive hemoglobin, or cases in which hemoglobin is not transporting oxygen well. Okay. So let's take a quick review about the previous ideas. So in this example here in A, we have 4 hemoglobin molecules. All of them are bound to oxygen, and all the binding sites are occupied. Therefore, we can say that we have a saturation of 100%. Okay. Okay, this is still odd stuff. Okay. The second picture here shows that we have a 50% anemia, and but all the remaining binding sites are occupied. Remember when I explained the example about the 6 hemoglobin molecules and 3 of them were destroyed, but saturation was still 100, but concentration was half. This is exactly the same thing, but now instead of my example of 1 hemoglobin can only bind 1 molecule, now it can normally bind 4. Okay. Okay. So here in this example here, in case of 50% anemia, we can see that we have 100% saturation, but we have a concentration that is halved. Now, if you count the molecules, here we have 16, but here we have only 8 molecules. So in case of 50% anemia, we are going to have a decrease in concentration by half, but saturation is going to still be 100. Okay. The last case is in the case of binding of carbon monoxide. First of all, carboxyhemoglobin is basically hemoglobin that's bound to carbon monoxide. So carbon monoxide has a very, very high affinity to hemoglobin. So most of the times, always, carbon monoxide is going to bind in the place of oxygen. Fortunately, we don't have a lot of carbon monoxide in our bodies. Okay. So carbon monoxide has a very high affinity to the binding sites of O2. Therefore, it's going to take its place. Now, although we have like all the hemoglobin is normal and you don't have an anemia, we have a decrease of concentration by 50%, because only half of the binding sites are occupied by oxygen. The remaining binding sites are occupied by what? By carbon monoxide. What about saturation? We can see that the binding sites here still exist, and hypothetically speaking, O2 can still bind to them. But because of carbon monoxide, O2 is not able to bind. But they are calculated in the percentage of saturation. Therefore, you are going to have a 50% saturation. Okay. This might not seem clear, so now we're going to explain it on the graph, and I hope it's going to be more clear, inshallah. Okay. So here in the graph, first of all, we can see that this is a concentration graph in relation to partial pressure. Okay. The orange graph here is the normal blood. Okay. It's basically here A, where we have normal erythrocytes and we have 100% saturation. All binding sites are occupied. Okay. The second graph here, which is the blue graph, is in the case of a 50% anemia. This is basically just like this one, okay, where we have 50% anemia, the saturation is 100%. Again, it is a concentration graph, and concentration in the case of 50% anemia is going to be halved, okay? Therefore, you're going to see that concentration has decreased from 200 in the normal case to 100 here, okay? The third graph is the green graph, and it's the one that's associated with the carbon monoxide binding in the place of oxygen. So in this example here, you can see that also concentration is halved and saturation is halved. Again, this is a concentration graph, so we're only concerned with concentration right now. You might assume that because the one that's caused by 50% anemia and the one that's caused by carbon monoxide binding both have 50% concentration, you might assume that both of them should have the same graph or the same shape of the graph. However, you can see that the green graph is a little bit shifted to the left of the blue graph. And what did we say shift to the left means? It means that there is an increase in affinity. But Abdul, you just said that when CO binds, it's going to take the place of O2. How is there an increase in affinity? That basically happens by when we have a hemoglobin molecule, and let's just say it has 4 binding sites and 2 are occupied with O2, with carbon monoxide. The remaining O2 binding sites are going to have a higher affinity to oxygen. That's the effect of carbon monoxide on hemoglobin itself. Again, so when carbon monoxide binds to a hemoglobin molecule, it's going to cause an increase in the affinity in the remaining 3 binding sites to O2. So these are basically 3 binding sites. Let me just change the color or something. Okay. So again, so this is a hemoglobin molecule, and we have it's bound by 2 carbon monoxide molecules. Now you are going to have 2 free binding sites here, okay, that are not occupied by anything. But because CO2 was bound to the hemoglobin, the affinity of these free sites is going to increase, and these sites are going to have a higher affinity to carbon monoxide. Okay. Okay. So basically, now, you might think of this as a good effect, but actually it's a bad effect. Let's think about a hemoglobin that's trying to deliver blood to a tissue that's in need of oxygen. When hemoglobin comes here, but unfortunately, it's occupied by both carbon monoxide, by 2 carbon monoxide ions, and by 2 carbon monoxide molecules and 2 oxygen molecules. Now, and the hemoglobin wants to drop these oxygen molecules to the tissue. However, because due to the effect of the carbon monoxide on the hemoglobin, this hemoglobin won't be able to release these O2 molecules. Why? Because it has now a higher affinity to these molecules. Is this idea clear? Okay, tamam. Okay, so in general, carbon monoxide has 2 bad effects on hemoglobin. The first one, it's going to bind in the place of O2, therefore it's going to decrease saturation. The second one is that it's going to increase the affinity of the other binding sites to O2. Okay. Therefore, it is going to cause the hemoglobin not to be able to release the O2 to the tissue. Okay, it's not good for two reasons. Okay, the first one is that it's going to it's going to bind in the place of O2 on the hemoglobin. Therefore, you are going to have a decrease in saturation. Okay, the second reason is that okay, so let's say again, say this is the hemoglobin molecules. Now, okay, now we are going to have two carbon monoxide ions that are bound in the place of oxygen. We still have two empty slots for O2 to bind. Okay, these empty slots, because CO2 was bound to these slots above, the empty slots now are going to have a higher affinity to oxygen. So oxygen is going to bind here with a higher affinity. Are we good till now? Okay. Now, when hemoglobin is trying to deliver blood, sorry, when hemoglobin is trying to deliver oxygen to a tissue. So this is a hemoglobin molecule. This is the same hemoglobin molecule, and this is a tissue. When hemoglobin wants to drop this oxygen back to the tissue, okay, it won't be able to do so. Why does that happen? It happens because it's the CO that's bound to it is going to increase its affinity to oxygen, and now oxygen cannot unbind from its place. Again, because CO has a high affinity, will cause an increase in affinity to oxygen. Now oxygen cannot leave its place. It cannot unbind the hemoglobin and diffuse to the other side. That's basically the bad effect of carbon monoxide on O2 delivery to the organs. Okay. By the way, Dr. Hashim really explained this in a very good example. He said that when you are in a bus and we have like 50% are bad guys and 50% are good guys, okay? And when these good guys wants to go down from the bus to their destination, the bad guys are going to stop them. The bad guys in this example are the carbon monoxide molecules, and the good guys are the O2 that are trying to diffuse down. Okay, so let's go to the next part. So this is we said that we have two scenarios in which O2, in which hemoglobin is not well functional. The first one is in case of carboxyhemoglobin, or when it's bound by carbon monoxide. The second case is in the case of oxidation. So in a normal cycle, you would see that deoxyhemoglobin, which is the free hemoglobin that's not bound to O2, okay, deoxy, no O2, it's going to undergo the process of oxygenation, or basically the addition of an O2 molecule, okay? In that case, it's going to become oxyhemoglobin. And oxyhemoglobin is good, is going to move in the body and is going to transport O2 to the needing organs, okay? At the organs level, is going to the oxyhemoglobin is going to undergo deoxygenation, and is going to come back to its original format, or the deoxyhemoglobin. Okay, this is a normal cycle. It happens all the time in our body. Okay, now the second cycle here is the cycle of oxidation. In the case of oxidation here, deoxyhemoglobin is going to undergo oxidation due to some oxidizing substances. Okay, you don't have to know any example of oxidizing substances. You only have to know the concept. Okay, so deoxyhemoglobin here is going to undergo oxidation, and it's going to become something called methemoglobin. This methemoglobin is a bad hemoglobin. It cannot bind any O2. Therefore, it's useless in the transport of O2 from the lungs to the organs. Okay? So, at normal conditions, this methemoglobin is going to undergo reduction by an enzyme called methemoglobin reductase, and it's going to go back to the deoxyhemoglobin format. Okay? However, in some conditions in which the methemoglobin reductase enzyme is not well functioning, the hemoglobin is going to get stuck in the methemoglobin format, and it's going to be useless. It cannot transport oxygen in this format. Okay? Okay, so are we all good until now? Do we have any questions or do you want me to repeat anything? Okay, great. I guess we're good. Come on. Okay. Now we are done with the O2 cycle, with the O2 transportation. Let's discuss now about CO2. Okay. So basically CO2 here, CO2 has three forms of transportation. The first one is the physically dissolved form. Basically, this is similar to the O2 form, physically dissolved form of O2 that we just discussed. It's basically free CO2 ions, molecules that are flowing freely in the blood. Okay? Okay. Carbon dioxide also has two chemical forms of getting transported. The first one is bicarbonate, and the second one is carbamate. Carbamate is also known as carbamino compounds. Okay, so most of the CO2 that's transported in our bodies is transported in the form of bicarbonate. Okay, 10% is transported as physically dissolved CO2, and the remaining are transported as carbamate. Okay. So physically dissolved CO2 is very simple. Just basically physically dissolved, just like the O2 that we just discussed. Okay. Um, what about bicarbonate and carbamate? So let's start first of all with bicarbonate. So basically, CO2 from the organs is going to diffuse to the erythrocytes and plasma. CO2 is produced in the organs due to its normal functions, such as like the Krebs cycle and the like fatty acids and these things. Okay, so CO2 is going to be produced and it's going to diffuse to the plasma and the erythrocytes. In the plasma and in the erythrocytes, the CO2 is going to interact with water. Water is present in both, okay? And due to this interaction, they are going to produce both bicarbonate and H plus ions, okay? This reaction occurs in both the plasma and in the RBCs, okay? However, it happens at a higher rate in the RBCs. Why does that happen? It happens because of two reasons. The reaction goes faster in RBCs because of two reasons. The first one is the presence of the enzyme carboanhydrase. This enzyme is present in red blood cells, but it's not present in the plasma. Okay. The second reason why this reaction would go faster inside the erythrocytes is due to the buffering of H plus by hemoglobin. So what basically happens is that hemoglobin inside the erythrocytes is going to take up that H plus ions. And when these H plus ions are removed, this is going to push the reaction in this direction. It's going to accelerate it so that it's going to go faster. And when this reaction is going to go faster, more HCO3 minus is going to be produced inside the erythrocytes than in the plasma. Okay. When that happens and we have more HCO3 minus inside the red blood cells, than in the plasma, then SCO3 minus is going to have a pressure gradient and it's going to start diffusing from the red blood cells to the plasma in exchange with chlorine. Okay. This exchange is going to keep continuing up until we have an equal amount of bicarbonate in both the plasma and the erythrocytes. Okay. What about the carbamate? Okay, for carbamate, the same CO2 that's diffusing from the tissue, instead of reacting with water this time, is going to react with, or it's going to combine with proteins or blood proteins, mainly hemoglobin. Okay, it's going to mainly combine with hemoglobin. When it does that, it's going to produce something called carbamino compounds or carbamate. So basically, carbamate is a carbon molecule that's bound to the N terminal of the amino terminal of the blood proteins, which is most of the time hemoglobin. Okay. Do I have to repeat anything about this? Bicarbonate or carbamate? Okay, come on. Now let's take a look at the CO2 dissociation curve. Just like the O2 has a dissociation curve, CO2 has one. Now we see that this graph shows partial pressure in relation to CO2 concentration. Okay. As you can see here, we have a dissolved form, which is the normal CO2. And the difference between these two graphs shows the chemical form, which is both bicarbonate and carbamate. Okay, someone wants me to repeat carbamate. Okay, I'm going to do that. Okay, again. So what happens here is that CO2 is going to come, but instead of reacting with water to produce bicarbonate, now it's going to react with blood proteins, mainly hemoglobin. Okay, when it does that, it's going to produce something called a carbamino compound. A carbamino compound is basically a CO2 that's bound to the N terminal of a protein. Okay. Sorry, it's going to be the carbamino compound is a CO2 that's basically bound to the terminals to the amino acids of a blood protein. In this example here, it's hemoglobin. Most of the time, it's hemoglobin too. So you can forget about other proteins. Just think of it like this way: CO2 is going to interact with hemoglobin and is going to produce what? A carbamino compound. Carbamino compound is a CO2 that is bound to to the amino acids of a certain protein. Okay. Okay. Tamam. Again, this graph shows both the dissolved CO2 and the chemical form. The chemical form includes both the bicarbonate and the carbamate. Okay. As you can see here, the difference between the arterial concentration of CO2 and mixed venous concentration of CO2 is about 43 milliliter for each liter of blood. Okay. However, when this change in concentration happens, we only see a decrease or an increase of 5 millimeter mercury of pressure. When you remember back in the, sorry, I should have explained something, but I forgot about it. I'm going to re-explain it now. As you can see here in the O2 dissociation curve, as you can see in the O2 dissociation curve, you can see that, okay, you can see that at the very top here, we have a plateau, okay? This plateau is very important. Why is it important? It's important because in cases of hypoxia, for example, when you go up high in the mountains, okay, in case of hypoxia, you are going to see a decrease of PO2 in the environment. When that decrease of PO2 happens, let's say from 100 to about 60, okay, when that decrease of PO2 happens, the saturation is only going to be affected by about 5 to 10%. And the body can still live with a 90% saturation. Okay, this can still provide enough oxygenation to the organs. Okay, so again, why is this plateau important? It's important because when there's a decrease in PO2 for whatever reason in the environment, the oxygen saturation is not going to be affected that much. At the organ levels here between 0.5 and 0.7, we can see that the graph is a little bit steep. Okay, so this steep graph. So what happens here is that when hemoglobin is giving out O2 to the organs, it is going to lose some saturation. So let's say saturation might drop from 0.75 to 0.5, for example, okay? However, this drop of saturation is only associated with what? About 10 to 13 like a decrease of PO2 with only a 10 to 13 millimeter mercuries. Again, when at the organ levels, the hemoglobin is going to drop off oxygen. When that happens, saturation is going to decrease. However, this decrease in saturation is only associated with a small decrease in partial pressure. Why does the body wants to maintain this amount of partial pressure? It wants to maintain it mainly to maintain the pressure difference, so that O2 will keep diffusing from the blood to the organs. Okay? We good? تمام. Okay. Okay. تمام. So if we come back here to the CO2 graph, as you can see here, although we have a very large drop in saturation here or in concentration here, there's a very small decrease in partial pressure. This basically happens because the graph here is it's steep, okay? Just like when we explain the organ level at the O2 dissociation curve. This is the O2 dissociation curve. Okay, here is the organ level. As you can see, this part is steep. So although there is a decrease in concentration, the partial pressure will still be high. Okay, here is similar to that. However, if you remember when you said that at the plateau level, if we have a decrease in concentration here at the plateau level, we're going to see a very large decrease in PO2. That's because we have a plateau here. But the graph here is steep. What mainly you have to know here is that the graph of CO2 is too much steep that even a great decrease in CO2 concentration is only associated with a small decrease in partial pressure. Okay. Now we're going to discuss two factors that are going to cause shifts in the CO2 graph. So the first one is H plus. When H plus is added to a solution that has CO2 or bicarbonate, sorry, it's going to interact with this bicarbonate and it's going to produce what? H2O and CO2. Unfortunately, this form of CO2 is not like this bicarbonate. This bicarbonate can stay in the fluid and it's just going to chill there. However, the CO2 is going to escape. It's going to escape the fluid and it's going to cause a shift to the right. So when H plus is added, it's going to interact with HCO3 minus, which is a form of carbon dioxide, and it's going to convert it to CO2. This CO2 is going to escape the solution, and therefore we are going to see a right shift. Okay, this more looks like a shift downward, but that's due to the shape of the graph. Okay, actually it's a shift to the right. Okay. Okay. The second factor that's going to affect the dissociation curve of CO2 is oxygen. Remember when we said that H plus and CO2 have an effect on hemoglobin that will cause a decrease in the O2 affinity, and that was known as the Bohr effect. Okay. Similarly, O2 has an effect on the hemoglobin. So what happens is that O2 is going to bind to the hemoglobin. When it binds to hemoglobin, it's going to decrease the hemoglobin's affinity to both H plus and CO2. And this is known as the Haldane effect. So what are the two factors that affect the CO2 binding curve? These are two. The first one is H plus, and the second one is O2. Okay, H plus is going to convert bicarbonate to CO2 that's going to escape, and O2 is going to decrease the affinity of hemoglobin or decrease the binding of CO2 and H plus to the hemoglobin. Okay. You have any questions before we practice some questions? Okay, no. Okay. Okay. Let's start with the first question. Here in the first questions, as you can see, in the hemoglobin O2 dissociation curve, as you can see here, we have a shift from graph A to graph B. What could be causing this shift of the graph? C. Okay. Okay, that's okay. Good. I'm glad you understand. Okay. So basically, C is the correct answer. Remember, what are the four factors that can cause a shift to the right? So this is again a shift to the right. What are the four factors that can cause a shift to the right? These are increase in CO2, increase in H plus, increase in biphosphoglycerate, and an increase in temperature. And you see that there is a mnemonic, cadet faces right. Okay. Let's go to the second question here. A 30-year-old woman moves with her family from New York City to Leavesdale, Colorado. Like it's above sea level by 10,020 feet, 200 feet, and it's very high. Okay. Which of the following will occur as the result of residing at high altitude? Yes. Okay, good. Remember when I said that BBG increases in cases of chronic hypoxia, such as going up high in the mountains? or in cases of chronic lung disease. So this is an example where BBG, okay, is going to increase. And what did we say happens when BBG increases? We are going to see a shift to the right. So good. Should I repeat that, or is it clear? I know you might not get that because you probably won't remember it, but when you study the lecture, it's going to be easy, inshallah. Okay, tamam. Now, question 3. An increase in which of the following increases the O2 affinity of hemoglobin? Focus, okay? An increase in which of the following increases the O2 affinity of hemoglobin? Mnemonic. Okay, I'm going to mention the mnemonic at the end. Okay, good, great. The correct answer is actually E. Again, we mentioned that although in the case of 50% anemia and in the case of 50% binding of carbon monoxide, we are going to see an O2 concentration that's half. But we can see that the CO, the carbon monoxide curve, is a shift to the left. Why does that happen? It happens because CO is going to increase the affinity of the remaining 3 binding sites. And this is the bad effect of CO on the O2 delivery to the organs. Now someone requested that I repeat the mnemonic, so I'm going to repeat it. So basically the mnemonic is, okay, we said that the first one is increase in carbon dioxide. The second one is an increase in H plus or a decrease in pH, okay? So we can see that C is a C, okay? We can just write C, okay? H plus or a decrease in pH means there is an acidity. So we can write an A. DBG or diphosphoglycerate or BBG, we're going to use the D for that, and it's a D, okay? So now until now we have C for carbon dioxide, A for acidity, and D for diphosphoglycerate. Then we have an E. This is basically for exercise. You don't have to know this for this lecture, but it's good to know, okay? It's good to know. What about the last one is temperature, and you can see the temperature here. We wrote the T for temperature, okay? So in case of an increase of carbon dioxide, in case of acidity, in case of an increase of DBG, in case of exercise, in case of an increase of temperature, we are going to see a shift to the right. So cadet, C-A-D-E-T, faces right. Cadet faces right means that when you have these factors, we are going to see a shift to the right or a decrease in affinity. Okay. Okay. Is this question clear for all now? Or should I repeat anything? The CO1? Tamam. Okay. Any questions? Okay, guys. Before ending this session, I really wanna wish you good luck in your exam. I know it's in 2 weeks, but I really advise you to use your time well. Practice questions. Practice as much questions as you can. The pulmonary questions can really be a lot tricky, and also cardio questions can also be tricky. So you have to focus and solve as questions as you can. Okay. So good luck in your exams, and thank you for your attendance, guys. Okay. I hope I can teach you more in the future, inshallah. Thank you.